Bismillahirrahmanirrahim. Today, inshallah, we'll talk about mitosis. This is a sufficiently important topic, I think, that we have to be methodical and not try to rush through. Once you understand it one time, I think, inshallah, it will be easier in the future to add details to this topic. So, mitosis, simplistically put, is replication of cell or duplication of cell, where one cell divides into two identical cells. So one cell kind of copies itself into two different cells. This is called cellular division. So mitosis is a process of cell division or cellular division. So this, in fact, I think is the formula for standard deviation. Anyways, now let's look at this process in the big picture wise. What you see before you is the simplistic big picture view of mitosis, where one cell divides into two cells. First thing that we have to understand is this cell is identical to the two daughter cells. The two new cells that are produced are called daughter cells, and they're identical to the parent cell. For that to happen, first thing you have to understand, the genetic material, the DNA of the parent cell has to duplicate itself, because if you notice in the end, there are two nuclei. So how do you generate an extra nucleus? Obviously, you have to first copy the genetic material, and then you have to separate that genetic material. Now, when a cell divides, the critical aspect of cell division is the nucleus. Okay, is authentic, faithful transmission, replication, and division of the nucleus. So the key aspect of cell division is dividing the one nucleus and making a faithful copy of it and dividing into two. The rest of the stuff, the cytoplasm and etc., like dividing into, is just like, just squishing into, pinching into two halves, so to speak, because cytoplasm, is water, enzymes which can be produced or imported from the outside. So that's not nearly as important aspect of cell division as the authentic duplication of the nucleus. So that's the key. So all the action, so to speak, of the cell division is occurring in the nucleus and aspects of the nucleus. So one thing that we have to understand is for cell division to occur, the genetic material has to be duplicated. And the process of cell division is, in fact, in summary, a authentic division of the cellular material into two different cells like this. This is a cartoon picture. It actually does not happen exactly like this, as you will see, inshallah. Now understand that cells don't always divide. Some cells don't divide, period. Like the nerve cells, that typically I give examples of some cells that once you're born, that's pretty much all the nerve cells that you have. You don't grow more brain cells, so to speak, right? Whereas some other cells, like skin cells, they divide constantly, yeah? So intestinal lining cells, for example, are cells that divide constantly. So there's some cells that divide infrequently and other cells that divide frequently. So this process is different for different cells. But in general, you can say that a cell has a lifespan, right? Just like a person is born, then they have a toddler age, and they have a young child age, and they have an adolescent age, and young teenager age, and young adult age, and then mature age, and etc. So that's the lifespan of a person. Similarly, a cell's life can be divided into ages or times, And the part of the cell where the cell is actually dividing, that phase of the cell is called the mitosis phase. The entire cycle of the life of a cell is called the cell cycle. The cell cycle is the life history, like the cell is born, it matures, and it divides, etc. So that's the cell cycle. And the cell cycle typically is divided into 4 parts, okay? Once a cell goes through mitosis, basically it's a new cell, right? Okay? And then it grows. So G1 is basically the growth of the cell. For example, in bone marrow, a person's bone marrow, a new white cell is born, and then after it's born, it kind of grows and matures, so to speak, right? And then one day it decides to, go undergo mitosis, okay? So it decides how does the cell decide? Obviously, that's a metaphorical term, but when cell is decided to go through a mitosis, okay? So now here's the cell. Now it's grown. Okay. Now, for it to go through cell division, the first thing it has to do is, as I said, it has to duplicate its DNA material. So there's a very long period, relatively to mitosis, a longer period of what's called the synthesis phase or the S phase, where the DNA is duplicated. You must understand, duplicating this DNA is remarkably accurate process, and we have like miles and thousands of miles of DNA, so to speak, literally, if you were to thread it out, and all has to be accurately duplicated. So the synthesis phase is actually, in fact, frequently much longer than the actual cell division phase. So this is the phase of the cell cycle where the DNA is being duplicated, so that now the new copy and the old copy can be separated out into making two different cells. So that's the synthesis phase. A lot happens in the synthesis phase other than just the DNA replication, but predominantly that's the big important aspect of the synthesis phase. And just as the synthesis phase is complete, there's a secondary preparation phase that occurs, so to speak. The right enzymes have to be produced, and other machinery has to be gotten ready, so to speak. And once the cell is finally ready to undergo through mitosis, and it goes through mitosis, making two different cells, and then the new cells go through the cell cycle again. So this is called the cell cycle. It starts with the growth phase, synthesis phase, G2 phase, which is further growth and preparation for mitosis, and finally mitosis phase. Okay, so that's the life cycle of a cell. Now here's another way of looking at the same cell cycle. In this case, you see the mitosis here, okay, mitosis G1 S phase and G2 phase. The mitosis itself is divided into different parts. So the mitosis is like the cell division phase. It has like Act 1, Act 2, Act 3, Act 4, so to speak. It's like a Shakespearean play in a way. And it has typically divided into these four different phases. They're called prophase, metaphase, anaphase, and telophase. So mitosis itself has very scheduled sets of events that are named, which we'll talk about. Okay? The phase that's between mitosis is typically given interphase as a term. Now look at this. This looks like some plant sample of cells. Now in this micrograph, light microscope, what you see here is cells at different stages of their life. Okay, when the cell is not dividing, it looks like this. This is nucleus and it's doing its thing. When the cell is dividing, you see chromosomes. Chromosomes are only seen when the cell division occurs, because chromosome is like wound up, tied up, rolled up DNA. It's kind of like if you have your clothes in the closet, and it might be all messed up in there, But if let's say if you had to pack them when you're going for a trip, you pack that all up in a suitcase. Now that's chromosomes. So chromosomes are like packaged up DNA. You only see chromosomes when the cell is being ready to divide. So you can see in this various cells over here, the different cells are at different stages of cellular division, and you can point out some of these, and this will become readily apparent to us once we have studied these, inshallah. So the first thing that has to happen is, the first thing is the DNA is replicated, right? So if you can imagine a cell nucleus, and then inside this nucleus is like a soup of DNA. It looks like it's like more like a spaghetti of DNA. A whole of that spaghetti is duplicated. So now you have like two spaghettis in that nuclei, and those spaghetti of DNAs. Okay, how are you gonna separate that? Can you imagine you have a bowl of soup with two giant spaghetti, a bowl with two giant spaghetti pieces all mixed up? How are you gonna separate that out, right? It's going to be very tough. So the same thing happened. The DNA in its natural form, open form, is very loose. It's a big mess of hair, so to speak. It's like that. So what happens is the DNA is coiled, and then coiled again, is coiled again, and very compactly packaged. And the end result is from going from a spaghetti nuclei to nucleus to what appears to be chromosomes. So this represents packaging of the chromatin, which is unwound free DNA, so to speak. And once it gets packaged, it becomes a chromosome, which becomes visible to the under the microscope, the light microscope here. So here's DNA, and the DNA gets, here you take like little proteins, and then you wound them like you wound on a spool, and then you package those further, and you package those further into chromosomes. Now look at this. Chromosomes has two of these, right? Because they're duplicated. This process of binding occurs after the chromosome has been duplicated. These are exact copies of one another, but they kind of hang in the middle over here. They're not separate copies, okay? So that's the first thing. So once the DNA is duplicated, it's wound, and it's a chromosome, a chromatin is converted into a chromosome. Now look at this. This illustrates the packaging of the DNA. So normal DNA is the double helix, looks like this. It's a beautiful representation. We did this, but God bless him. Just look at this. This beautiful representation. So DNA is unwound free DNA. That's the chromatin DNA. That's how it appears when the cell is not dividing. But when the cell is ready to divide, this DNA is packaged. You take like a spool, and DNA is like a thread, or you wind it around this spool. That spool is basically big chunk proteins, and you keep winding it. Let's see how you take that, and you wound the DNA, and then you take a whole bunch of spools, and you keep winding them into big, thick, like rope structures. So becomes like a big ropey thing. You see what's happening here? So you take DNA and you wind it, and you're producing into a chromosome. So this shows you the transformation of chromatin, freed of unwound DNA, to a chromosome like this. Okay. This is one chromosome, and this is duplicated chromosome over here on this side. Okay. Now the chromosome has its own anatomy. It has typically a short arm, the short side, and the long side is typically called the long arm. Short arm P, long arm is Q, like PQ. P's and Q's, right? And when they're joined together, that's called a centromere. Okay. Now, mind you, this is the original DNA, perhaps, and this is the replicated DNA that will belong in the future in the new cell that is being produced. So this is one DNA, and this is the copy of the DNA that is still hanging on over here. They're connected at the centromere because you want to have organized separation of this DNA so that the both of these DNA don't end up in one cell or something. So this demonstrates the process of condensation of chromatin, which is the free DNA, unwound DNA, into chromosome. Now look at this. Now this is this is the chromosome, and here it's paired with its duplicated chromosome. So originally the chromosome, let's say this was this original chromosome, and it got duplicated before it became a chromosome, and now here are the two identical chromosomes, and they're attached to each other in the middle. That's called the centromere. Now there's some interesting aspects of biology here. Biology is said is the only place where division is the same as multiplication, is cellular division and cellular multiplication, right? So and it's also very curiously, this terminology is very funny. So this is one chromosome. Okay. Now when it's here with duplicated chromosomes right here, this whole thing is still called one chromosome. This is one, but there are two chromosomes inside of it. That's one chromosome. That's one chromosome. But the whole thing is also called one chromosome. It's also one place possibly in science where 1 equals 2. So anyway, so it's kind of curious, but it's important to understand that this is one chromosome, this is one chromosome, and this whole thing is also one chromosome. Now these individual chromosomes, they're given names. They're called chromatids, sister chromatids. Sister meaning there are copies of each other. Sister chromatids. So two sister chromatids make up one chromosome, and each sister chromatids is a chromosome in itself. Go figure. But that's how it is. Now, to make sure you appreciate the point, Here it is. This is an illustration of aspect of mitosis, which we'll study in just a second. Now, here's the cells dividing. Here's a chromosome with the 2 sister chromatids. In this picture, see, you see 4 chromosomes. This cell has 4 chromosomes. 1, 2, 3, 4. Now, this cell, once it's dividing, see that they have separated from each other. It's dividing. It has 8 chromosomes, because each one of them individually is called a chromosome. Okay, that's just the terminology peculiarity, which might confuse some people, so for clarification purposes. Okay, now we're going to talk about the actual process of mitosis. So mitosis is a process of cell division, and as suggested, the key aspect of cellular division is authentic duplication and transmission of the genetic material. Now, the mitosis is typically divided into 4 phases. Okay, I still remember from my biology class in 9th grade, you can remember that by PMAT, P-M-A-T, those 4 phases, okay, PMAT. They are prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, telophase. After a while, these become quite natural. PMAT, prophase, metaphase, anaphase, and telophase. And some very different, unique things happen in each individual phases, okay? Now, I'm going to describe them briefly, and I'm going to elaborate on each one of them slightly before, as we go along. Now, notice in prophase, you have already made a copy of the DNA and has been condensed into chromosome. So by the time this prophase starts over here, the DNA already has been replicated, but because that happens in the S phase. And then by the time prophase is started, the DNA has also been condensed into chromosome. Okay, so look where we are. So prophase is the beginning of the mitosis. So by the beginning of the mitosis, this has already occurred. Okay. Now, if you allow me, look at this picture over here. That's kind of like the middle, the act 3, the climax of the mitosis, if you like. Okay, in this picture, what's happening is, okay, all the chromosomes are lined up in the middle, or religiously lined up in the middle, so to speak. Okay, why? Because the cell is going to divide it like right across over here, and one of these duplicated chromosomes needs to end up over there, the other one needs to end up over here like this, and then the cell can divide over here now that you have 2 cells. So the lining up of the DNA is kind of like the thrust of mitosis. That's where you are going. So the goal of mitosis is to reach the climax, which is lining up of all the chromosomes in the middle, and then so each can be separated equally. Because if they don't line up properly, haphazardly, there might be a mix-up in which one goes where. So they all have to line up perfectly like this, so that one goes this way, one goes this way, one goes this way, one goes that way. So that's like the goal climax of the mitosis. For this to happen, observe what needs to be. They have to line up, right? So, and then for them to go to each side, the nucleus. Here's a nucleus. The nuclear membrane has to break, because where's the DNA? The DNA is inside the nucleus. So the nucleus, the membrane, the nuclear membrane, not the cell membrane, the nuclear membrane has to dissolve. It has to go away. It has to disappear, because how else are they going to go to 2 different sides, and then the cell is going to divide? So one of the things that has to go from here to here is the nuclear membrane that you see over here has to dissolve, right? Otherwise, there's no going to be dividing in 2 halves. The second thing that has to happen is, you see these spider-like thingies, like spider webs. These spider webs have to form, and these spider webs are generated by these little funky little thingies that are over here on each side. They're called centrioles. So these spider webs are generated by these structures called centrioles. So what they really do is they're like chandelier type things from each side of the cell, and they put out webs, literally like Spider-Man puts out his webs, and they go in there and they capture these. So these chandelier type things, they capture the centromeres over here, and then they are actually the one that are pulling, and they really break these two apart, like one, so that half of them go over there, half of them go over here. So the two key aspects of the mitosis is, one, that all the chromosomes have to line up in the middle, and the spindle web, which is what this is, the spindle web, the spider web, so to speak, they have to form, and the nucleus membrane has to disappear. Right? So look, that's what happens between prophase and this phase, which is the metaphase. The nucleus over here dissolves, it's gone over here, and now the spindle is forming over here. Now look at this. This is quite remarkable. Now, once the nucleus breaks, these things have a tendency to go here and there, right? You follow? They just kind of like, like, oh, I'm free, I'm like diffuse over here, osmosis, no. So these spindle fibers actually are the one who are controlling everything. So they grab onto this egg spider and they bring them on over here. So the dissolution of the nuclear membrane, okay, occurs, and the formation of the spindle fiber occurs in the prophase, the first phase of mitosis. Okay, look at this over here. You can still see this all the chromosome here, and they're coming over here, and in the process of once that is accomplished, now the accurate lining up of the mitosis occurs in metaphase. So, okay, middle. So metaphase as a metaphase is like they line up in the middle. Metaphase, they line up in the middle. So perfect lining up of the chromosomes occurs in the metaphase. All the work that goes into making that perfect lineup occurs like here in the prophase. Once they have lined up, okay, each sister chromatid, each of the duplicated chromosomes, each sister chromatid is separated in this phase, and that phase is called the anaphase. Once that is completed, once the duplicated DNA is authentically, accurately, faithfully separated from another, see, right? Now you can just divide the cell into two, and now you have two new cells. Right? So once you divide the cell like this, for example, the nuclear membrane has to reappear because that DNA that's there has to be protected. The nuclear membrane has to reappear, and the chromosome have to dissolve into chromatin, the natural state. Right? So when the cells, chromosomes separate into sister chromatid, this phase is called anaphase. I remember this by they move away from each other. Anaphase. They move away from each other. Anaphase. And in telophase, the cell divides into two, the nuclear membrane reappears, and the chromosome goes back into its natural state, the chromatin state. As a review, watch this, okay? In the prophase, the nuclear membrane dissolves, the chromosomes begin to line up with the help of the spindle fibers. And what's controlling the spindle fiber are these very curious and beautiful structures called centrioles over here, and they're the ones that are generating the spider web type web over here, and they line the chromosomes up in the middle, in the metaphase, in the middle, and the anaphase, the sister chromatids separate into two different to the two poles, and in the telophase, the last phase, the cell divides. The cytokinesis occurs, division of the cell, cytokinesis. The actual cell pinches into two, this pinching of the cell, and reappearance of the membrane, and the dissolution of the chromosome into the normal chromatin state. So that's the summary of chromosome. It's worth it to take time to appreciate this show. Now, this is the same thing in which I have described already. Now we're going to look at these phases in more detail, and you'll appreciate it, I think. Now, this is the synthesis phase. As you can see, the chromosome is unwound. It's in the chromatin phase. It's like the spaghetti phase, because how are you going to make a duplicate copy of the DNA once you made a chromosome out of it, right? It's going to be impossible. So the duplication of DNA has to occur when it's unwound in the chromatin state. So in the synthesis phase, the DNA is duplicated. Many other things have to be duplicated. You have to make other proteins, etc. Whatever the proteins you're going to need for the cell division itself. So that occurs in the synthesis phase. Now look at the prophase, the first phase of the mitosis. In this case, the spindle fiber is being formed. The spindle web is being formed. And these centrioles, they migrate. See, they're right here, right? They're next to each other. But then they migrate to opposite poles of the cells. So the centrioles, those beautiful yellow things over here, I like them a lot because they have a very, very geometric, very mathematical orientation. The more you study them, the more you appreciate how this all cell division occurs. But anyways, those centrioles, the beautiful things, they migrate to opposite poles and they shoot out their webs. And notice also how the nuclear membrane is disappearing. So this is prophase, the spindle cells migrating and shooting out the spindle webs, and the chromosomes are beginning to line up in the middle, and the nuclear membrane is disappearing. Okay. Now, if you observe carefully this picture, it's very interesting the way they did this. Now, look at what happens to the individual code. They're kind of flying all over the place, right? It's like, oh, it's like, wait. It's like Spider-Man trying to rescue somebody. He's trying to grab them by the spindle. But otherwise, they might fly up to some weird corner of the cell or something like that. Yeah? All right. So that's the prophase. Now, in the metaphase, they're all lined up in the middle. So the accurate might now they grab the spindle fibers. They have grabbed them, right? Once they grab them, once they have grabbed the chromosomes, then bring them into the middle like that. They all line up in the middle. It's beautiful. Once the spindle fibers have actually got the spider webs have caught the chromosomes, they bring them into the middle and line them up. So they line up in the middle of the cell like this, the metaphase plate. So this, when they line up, this thing highlights in the middle. See, like right there. See how that light? That's the metaphase. That's where the cell is going to divide. See, that's like that. That's called the metaphase plate. Now, in the anaphase is when they move away from each other. The sister chromatids divide. They separate from each other, and they actually move into the opposite poles of the cells. They move into opposite poles of the cells. That's the anaphase. And finally, in the telophase, look what happens. The cell divides into two. The cell pinches into two, right? The cell actually pinches into two. The chromosomes dissolve into the chromatin, and the nuclear membrane reforms to protect the DNA. That's the telophase. Prophase, metaphase, anaphase, and telophase. Now, I just want to bring this to attention, the big picture again. This is the synthesis phase. DNA is replicated in the prophase, right? The spindle fiber forms, the centrioles move to the opposite side, the nuclear membrane disappears, and the centrioles shoot out their spider webs, and they catch it. In the metaphase, all the chromosomes are lined up in the middle. Now, this is a beautiful cartoon representation how the spindle fibers go and capture the chromosome. See? Boom, like Spider-Man, like spider web. It's kind of cool, I think. See? Boom, they catch it. And then they bring it into the center, in the metaphase. And anaphase, sister chromatids separate. Come on, now sister chromatids separate. And in the telophase, the cell pinches or divides. In this process called cytokinesis, division of the cell, nuclear membrane reforms, and the chromosomes uncoil into chromatin. And the spindle fibers, of course, disappear. This is just a summary slide. As you can see, pretty, now it kind of makes sense, hopefully. Let's see that again. Excellent. Now here's another process. It's just kind of cool. People put the effort in making all this. So I hope you appreciate how in the beginning the DNA material is in chromatin form right here. It's duplicated and then it's turned into the chromosome form, right? Yeah, some of the details in these cartoon pictures are not accurate, I think, but we get the big picture, which is a very, very good. So now this is like time-lapse with microscopy. It's kind of cool. It's just, you can just see how a cell is dividing in, prophase, metaphase, anaphase, and telophase here. Okay? That's metaphase, anaphase, telophase. Pretty cool.
So that is actually mitosis in a brief overview. Inshallah, I hope you have opportunity to learn more about this process, which is quite beautiful. This is chromatin. This is chromosome. It's saying to get ready because mitosis starts in 5 minutes. It's all fine for your personal edification. Until next time then, salatu wassalamu ala rasulillah walhamdulillahi rabbil alameen. Assalamu alaikum.